Hello, everybody. Have you ever wondered what happens to proteins that we consume in the form of milk or eggs, dal, or meat for that matter? So, in today's video, we will try and understand what happens to these proteins, that is, how these proteins are digested in our human system. So, before we do so, we will try and understand a couple of terminologies that will help us in understanding the concept better. So the first is proteolytic enzymes. So proteolytic enzymes are enzymes that are involved in breaking down proteins. And interestingly, these proteolytic enzymes in our system are synthesized in inactive form. And these inactive form of protein enzymes, protein digestion enzymes, they are termed as zymogens. Being synthesized in inactive form, they have to be activated in our system. Now let's pay attention to some of the enzymes that are involved in protein digestion. So the first one is pepsin and followed by trypsin, chymotrypsin and carboxypeptidase A and B. As I mentioned that the proteolytic enzymes are secreted in inactive form which are called as zymogens. So here I have listed the inactive form, that is the zymogen forms of these enzymes. So pepsinogen has to be converted to pepsin. Similarly, trypsinogen into trypsin, chymotrypsinogen into chymotrypsin, procarboxypeptidase A and B into carboxypeptidase A and B. Now let's start understanding the digestion. Two types of digestion happens in our system. The first one is mechanical digestion and the second one is chemical digestion. So digestion starts in our oral cavity. The large food particles are broken down into smaller food particles with the help of the teeth present in our oral cavity. So, upon ingestion of protein-rich food, mechanical digestion, that is breaking down of larger protein particles into smaller particles, happens in our oral cavity. And the food travels down the esophagus and reaches stomach. So, in the stomach, we have cells called as parietal cells that secrete HCl. Similarly, there are another set of cells called as chief cells which secrete pepsinogen, which is one of the first enzymes that help in breaking down protein. So this pepsinogen is inactive, and in presence of HCl, it gets converted or gets activated into pepsin, and this pepsin helps in uncoiling the proteins present in our food. Upon uncoiling, the uncoiling of protein helps in further enzymatic digestion which happens in the intestine. As the food moves to our small intestine, that is the first part of small intestine called as duodenum. So here the chemical digestion of protein gets completed. Pancreas releases enzymes that further break down the larger protein molecules into smaller oligopeptides or dipeptides or tripeptides. Pancreas secretes trypsinogen, chymotrypsinogen and procarboxypeptidase A and B. These being in inactive form, they have to be converted to their active form so that these active form of enzymes can further break down the proteins. The mucosal intestinal cells secrete an enzyme called as enterokinase, which helps in activating trypsinogen into trypsin. And very interestingly, the activated trypsin can further help in activating trypsinogen into trypsin. And then this activated trypsin also helps in activating chymotrypsinogen into chymotrypsin. Similarly, procarboxypeptidase A and B into carboxypeptidase A and B. So these activated forms of the proteolytic enzymes breaks down the larger protein molecules into smaller protein molecules. Further, intestinal cells also make aminopeptidase, tripeptidase and dipeptidase. So these enzymes further help in breaking down the peptides into free amino acids or smaller peptides, usually di or tripeptides, which are eventually absorbed by the cells of the intestine, which are called as enterocytes, and via the enterocytes it reaches our portal circulation for further distribution. So far we understood protein digestion. Now let's understand how the protein digestion end products are absorbed in our intestine. So absorption happens in the duodenum, jejunum, as well as in the ileum portion of our intestine. So there are two major ways our body absorbs protein breakdown products. The first one being free amino acid transport, and then peptide transport using pepT1. Interestingly, our intestine absorbs di and tripeptides faster than free amino acids, thanks to a special transporter called as pepT1. Now, this transporter carries peptides along hydrogen ion concentration. As you can see, hydrogen ion, along with it, it carries di and tripeptides. Once inside the cell, these di and tripeptides undergo proteolysis, that is, they are further broken down into free amino acids. And these free amino acids enter our blood vessel. And remember, only free amino acids are allowed to leave the cell and then enter blood. Free amino acids enter the intestinal cells, that is enterocytes, using special carriers or transporters, as you can see here. So these carriers are specific for L form of amino acids. They are energy dependent. They also use sodium gradient, and then they work against concentration gradient. So as you can see, sodium gradient. So the entire process of protein digestion, in other words, the end product of protein digestion, uses ATP, that is energy, indirectly through a pump called the sodium ATPase, which maintains the sodium gradient. Absorption in the intestine does not depend only on amino acid transporters. It also depends on ion exchanges that creates the necessary gradient. So one of the most important among them is NHE3, that is sodium hydrogen exchanger 3. So as we understood, pepT1, that is peptide transporter 1, brings di and tripeptides into the enterocyte along with hydrogen ion, that is protons. For pepT1 to continue working, the cell must remove the excess hydrogen ion molecule, otherwise the gradient collapses. So that's exactly where NHE3 helps. So NHE3 removes hydrogen ions from the cell, and brings sodium ion. So NHE3 removes hydrogen ion from the cell, that is exports, and then brings in sodium ion in, thus maintaining the hydrogen ion gradient needed for pepT1 to keep functioning. Before we end, let's summarize very quickly the process of digestion and then absorption. So before absorption, protein must be broken down through digestion. In the stomach, pepsin begins breaking proteins into polypeptides. In the small intestine, pancreatic enzymes like trypsin, chymotrypsin, and carboxypeptidases further break them down into small peptides and amino acids. Only after digestion, absorption begins. So protein fragments are absorbed in two main forms: free amino acids through sodium-dependent transporters, di and tripeptides through the pepT1 transporter, which is faster. Inside the cell, peptides are converted into free amino acids, and only amino acids enter the blood stream. And pepT1 uses hydrogen ions to bring in peptides, and NHE3 removes excess hydrogen ion by exchanging it with sodium. So pepT1 absorbs peptides, and NHE3 keeps absorption going by maintaining the balance. Digestion prepares the protein, absorption delivers it to the body, and transporters like pepT1 and NHE3 ensures nothing is wasted. Hope this video was helpful. Thank you.